Good.
Hello, everyone. Welcome back to the third and last session in this series, the Spotlight in Rehabilitation series, showcasing the Brain Lab. Before I hand over the floor to Marie-Hélène Boudrias, I would like to read the land acknowledgement. McGill University is situated on the traditional territory of the Kanien'kehá:ka, a place which has long served as a site of meeting and exchange amongst nations. We recognize and respect the Kanien'kehá:ka as the traditional custodians of the lands and waters on which we meet today. Marie-Hélène, thank you for organizing this lovely set of lectures. We have three invited speakers today, and over to you. Thank you. Thank you all for being here.
Thank you to you as well. Bonjour à toutes et à tous. Merci de vous être de nouveau joints à nous en si grand nombre aujourd'hui dans le cadre de ce troisième et dernier webinaire sur le thème de la COVID. Before we begin in the presentation, please allow me to give you an overview of the research being carried out in my laboratory. Our research interest can be characterized into three areas. First, neuroimaging, where we quantify brain networks and identify biomarkers of brain function using the newest advances in neuroimaging and neurophysiological techniques, as well as signal processing. Today's first two presentations are related to this area of my research program. Second is neurorehabilitation, where we design therapeutic interventions specific to each individual based on brain biomarkers. We specifically focus on using brain stimulation and neurofeedback approach in the context of aging and stroke. And third, most recently, our team has been interested in the impact of COVID on the brain, the delivery of rehabilitation services, and the well-being of individuals impacted by long COVID. As we conclude today's discussion on COVID, I'd like to share an update of the current situation in Quebec. So this screenshot taken yesterday from the Association québécoise de l'encéphalomyélite myalgique provides recent data from COVID-19 Resources Canada, indicating that 1 in 31 people in Quebec is estimated to be currently infected with COVID-19. They also offer recommendation on the best way to protect ourselves from infection. Furthermore, according to the latest report from the Institut National de Santé Publique du Québec, the risk of developing long COVID increases with repeating infection, reaching 37% after a third one. So these are critical figures to keep in mind as we consider the future approach to manage infection-associated chronic disease. Today, we have the pleasure Today we will have the pleasure to hearing from Monserrat Casado, a PhD candidate in the integrated program in neuroscience at McGill University and a member of the Brain Lab since 2022. She will present the most recent result of her research project, which focus on identifying electroencephaly, EEG-based markers of long COVID symptoms. We are also delighted to welcome Professor Georgios Mitsis, an associate professor in the Department of Bioengineering and a William Dawson Scholar at McGill University. He has been an invaluable collaborator on numerous projects with the Brain Lab, including the co-supervision of graduate students. His research interests include mathematical modeling of physiological systems and multimodal functional neuroimaging. He has a particular interest in the function of autonomic centers in the brain, which, when dysregulated, can result in severe symptoms like POTS, postural orthostatic tachycardia syndrome. Today, he will discuss our ongoing study on dysautonomia in the context of long COVID. We are also thrilled to welcome Todd Davenport, professor and chair in the Department of Physical Therapy at the University of the Pacific in California. He is a scientific advisor for the WorkWell Foundation, whose research concerns a better understanding of fatigue and post-exertional malaise present in fatigue-related illness, such as myalgic encephalomyelitis, and long COVID. Professor Davenport also served as the chair of Long COVID Physio, an international association focused on peer support education and advocacy, one of the first to offer reliable and accessible online information for individuals living with long COVID. Professor Davenport will tell us more today about the challenges we face when dealing with infection-associated chronic diseases. We are always open to collaboration and welcome feedback, so feel free to contact us through the Brain Lab link. We would love to hear from you. Nous répondrons à vos questions en français et en anglais à la fin des présentations. We will answer questions at the end of the presentation. Sans plus attendre, je laisse la parole à Monserrat Casado.
Hi, everyone. Thank you for that introduction. I'm going to start sharing my slides. So thank you for inviting me, and thank you all for joining today. What I'm going to be presenting is the study that I'm doing as part of my PhD, and it is about finding biomarkers of post-COVID-19 condition using EEG. So we know to this day that COVID-19 can affect a lot of systems in the body, including the brain. And we also know that about 15% of people who get COVID-19 will have symptoms that linger after the acute infection. And if the symptoms are still present 3 months after the acute infection, last for at least 2 months, and don't have any other explanation, according to the World Health Organization, this can be considered part of PCC, also known as long COVID. Now, along with a broad range of systems that can be affected, there's also a lot of symptoms that can be present in PCC. And for this study, we decided to focus on fatigue, PEM, and brain fog, not only because they're some of the most common and persistent symptoms of PCC, but also because they have a big impact in people's quality of life. And importantly, they also suggest the involvement of the brain. Some studies have already found that there are brain alterations that appear to be related to these PCC symptoms, such as a reduction in cortical thickness, which is thought to be associated to brain fog. And also there's a study that used the TMS that found a reduced excitability of the motor cortex that appears to be associated with fatigue in PCC. So the same group that did the TMS study that I just mentioned published this new study a couple of months ago where they used TMS again, but this time they paired it with EEG to observe the activity of M1 and SMA of individuals with PCC who had fatigue, and they compared it to age-matched healthy controls. So what they found was that post-COVID fatigue is associated with a reduction in TMS-evoked beta oscillatory activity both in M1 and in SMA. And these findings are very relevant to our study because they set a critical precedent in understanding the neurophysiological impact of PCC, particularly in relation to fatigue. So considering that, now I'll start explaining our study. The objective that we set was to identify neural correlates that are associated to the main symptoms of PCC, mainly fatigue, brain fog, and PEM. And we are doing so by using EEG. This study is cross-sectional, and it has matched pairs, which means that we have a clinical group and we have a control group, which are paired in age and sex. And this study also has an exploratory component involving repeated measures, which I'll explain what it means in a second. So this is the sample size that we have. This is the final sample size, and the recruitment and data collection are finalized, and they were finalized in the last week of September. So a little less than 2 months ago, which means that this is still a work in progress, and I am not done. The results are not completely finalized, but I will talk about some of the results that are finalized to this point. So for the study, there were 2 main stages. The first one was done online, where participants filled out some clinical questionnaires and they resolved some cognitive tasks. And that was followed by an in-person appointment at our lab where we did a brief motor assessment and an EEG recording. I'll explain the stages more in depth, first starting with the online stage, during which participants filled out these questionnaires online and through the RedCap platform. And these are the questionnaires that we used to assess fatigue, PEM, and brain fog. So these were our main outcomes. And then after this, we also included these exploratory components for other important aspects such as quality of life, sleep, depression, anxiety, and comorbidities. And these are some of the things that we found there. So here we can see first a little bit of the demographics. Here's the sex distribution as well as the age distribution of both groups. This is for the PCC group, this is for the controls, and we have that only one participant in the PCC group was hospitalized during the COVID-19 infection. They needed oxygen supplementation, but they weren't admitted to the ICU. And then we have here the time since the COVID infection at the time of the EEG. And after this, we have the average scores of the questionnaire. So first we have the fatigue severity scale. For here, there's they have a threshold of 36, which means that any score 36 or higher is considered someone who presents severe fatigue. For the CFQ, the threshold was 43, so any score 43 or higher is considered a high score. And the DSQ-PEM was used to confirm the presence of PEM. So percentages here represent the participants who met the criteria for PEM. And then these are the next questionnaires, which were our exploratory components, where we can see that the PCC group has higher scores of daytime sleepiness, as well as anxiety, depression, comorbidities, and also more sleep disturbances and a lower score on quality of life. Then for the second part of the study that I talked about, which is the EEG recording, at the in-person lab appointment, we recorded brain signals for each participant. First, for the pre-task condition, which consisted of resting state recordings, but also 50 hand grips that were held for 4 seconds at 15% of maximum strength. And then after this baseline, participants had to do a visual motor learning task where they tracked some targets on the screen with a moving cursor by adjusting the force on the gripper. And they had 5 blocks of 20 trials for this task, which scores were recorded. And this task was meant to act as a cognitive effort so that afterwards we could repeat the baseline recordings to have post-task recordings to compare. If you remember, I mentioned an exploratory component involving repeated measures. So this is a repeated measure, and this is the exploratory component. And the way that we're analyzing all of this, first, from the resting state, we're analyzing the brain network with graph theory, functional connectivity, and complexity measures, which gives us an insight into how brain regions are interacting, if the network is stable, if it's able to adapt. And then from the hand grip recordings, which is what I will be focusing on, which are the results that we have more finalized, we're focusing on the motor cortex. And we're looking specifically at how the beta band power changes during movement. This includes the MRBD, which is a beta desynchronization that happens during movement. If you remember, I said the hand grips were held for 4 seconds. So this is what happens in the beta band during that time. It desynchronizes, it loses power, and this is called MRBD. And then after this, we have a rebound, which is once the movement stops, the beta band goes back to baseline or it goes a little bit higher. So that's the PMBR. Now I'll show some of the results that we have from that so far. First of all, there's different ways in which we can observe the beta oscillations during movement. One is to track the relative changes in power in relation to a normalized baseline. So this is the relative MRBD and PMBR. And in other words, we're normalizing here the baseline to 0, and we're tracking and we're calculating how much this power is moving during the movement stage, so the MRBD, and after movement for the PMBR. So far, the PMBR has shown to be significantly lower in the PCC group in electrode C3. So this is right here on top of the motor cortex, and it's assumed to represent the right motor hand area. This rebound is thought to reflect the reestablishment of the motor network's resting state and is associated with motor learning and sensory feedback processing, as well as the inhibition of further movement. So a reduced PMBR could reflect some ongoing dysfunction in motor control processes and cortical inhibition. And additionally, this lower PMBR is also correlated with high scores on the cognitive failures questionnaires. So this means that the reduced PMBR might not only reflect motor-related issues, but it could also be linked to some of the cognitive difficulties that are experienced by individuals with PCC. Another way to analyze the beta band in relation to movement is to look at the absolute power without any normalization. So this is the raw power. We're looking at how the power is without any normalization in the baseline. And we can see here the pre-movement stage, the movement stage, and the post-movement stage. So when we looked at this, we saw that on electrode C5, which is also here on the motor cortex, we found that during the movement period, which is the same as what we would be looking at in the relative MRBD, the PCC group has a higher beta power compared to the controls. And we're also analyzing the behavioral data that we have from this study, such as the reaction time from the gripping task. So this showed to be significantly higher in the PCC group, meaning that they were slower to react than the controls when squeezing the gripper during the 50 hand grips. This is one of the multiple trends that we have found, and we're studying to find out if they have any correlation with the biomarkers that we're finding through the EEG analysis. And to wrap everything up, the results that we've found so far, like the lower PMBR and the higher power during movement, the higher beta power during movement, these are patterns that have been previously linked to age-related changes in brain function, which suggests that PCC may impact the brain in ways that resemble aging. So identifying these patterns as potential biomarkers could help with detection, monitoring, and also to guiding interventions for PCC. To this day, there's multiple pharmacological and non-pharmacological interventions that exist, such as brain stimulation, which is actually something that we study at our lab, which have been proven to be successful at modulating the oscillatory properties of the brain and could potentially improve brain function and aid recovery in PCC. And last but not least, always a huge thank you to everybody who makes this possible. And thank you all for listening and for joining us today.
Okay, so yeah, thank you again, Marie-Hélène, and thanks to the School of Physical and Occupational Therapy for the invitation. And it's great to see it's very well attended too. So I think it is an issue that has generated attention, and for good reason. And as Marie-Hélène mentioned, yeah, there are some alarming trends in the occurrence of the prevalence of long COVID. So I think we need to understand this better and figure out how to treat it better depending on the phenotype of the individual, right? So today I'm going to be talking basically about what we are doing in our project that is related to post-COVID condition or long COVID, which we are trying to focus on its effects on the autonomic system, which are a common denominating factor in many of the symptoms, I think, and which is also supported by evidence. And we're going to try to do that using physiological monitoring and state-of-the-art neuroimaging. So I'll try to give you a general idea within the limited time that we have. So Monserrat gave a good introduction to post-COVID syndrome or post-COVID condition or post-COVID syndrome or long COVID. And you are probably aware that it affects many organs. And I think that the common denominating factor, as I mentioned, is that the autonomic nervous system seems to have a lot to do with it. So you will see that in many of these studies that have been published in the literature, that a lot of the symptoms that have been reported are directly related to the function of the autonomic nervous system. And that includes, for instance, orthostatic intolerance. It includes tachycardia. It includes breathlessness and even fatigue, which is in general related to the function of the autonomic nervous system. So there seems to be a common factor here that seems to relate to the autonomic system. Now, of course, the brain has also attracted a lot of attention in terms of damage that is possibly inflicted by COVID and post-COVID condition. And I will give a few examples here. For instance, one of the most high-profile studies that has been published until now was based on a large number of subjects from the UK Biobank, which were scanned before and after the COVID-19 outbreak. So what they did is that they used structural imaging, and they revealed a quite significant loss of gray matter on an average of 2%, which was correlated to cognition. And this reduction in gray matter was actually found to be present both in subjects that were hospitalized and non-hospitalized. So that didn't seem to be a major factor influencing the final outcome in terms of brain structure. And here you can see on this slide, yeah, So you can see the areas which were most significantly affected. Now, an important an important result from that study is that they talked about the brainstem too, which, as you may know, is the main hub of the autonomic control centers in the brain. So they did find some gray matter volume reduction in the brainstem, but the UK Biobank protocol is not optimized for the brainstem, so they could not go into too much further detail with regards to damage specifically in the brainstem. However, a more recent study, which was just published, did exactly that, and they used 7 Tesla structural MRI to more specifically examine the effects of post-COVID condition on the brainstem. And here you can see some results. And indeed, what they used is another way of obtaining MR data, which is called susceptibility mapping, which can detect in general microbleeds, microlesions, or effects of inflammation. So they did show that in a cohort of 30 hospitalized COVID-19 patients and 51 age-matched control, that indeed there was brainstem-specific damage that was centered around these 2 clusters that I'm showing on this slide. So, and they did find overall an increased MR susceptibility, which suggests some kind of damage in the brainstem, in the medulla, pons, and in general in the brainstem. This is another study which also Monse used in her presentation, so I won't go into too much detail here. Again, this study found out that there was a reduction in cortical thickness, which was correlated to the emergence of brain fog in post-COVID conditions. So again, further evidence that the brain is affected, and more specifically that the brainstem is affected. Now, a common factor for all these, for most of the studies that have been done until now, is that they focused on structural imaging. So there's way fewer functional imaging studies, and this is a gap that we're trying to fill with what we're trying to do. But I'll come to that a bit later. Now, if we come to autonomic dysfunction, of course, as you would imagine, people have started looking into that. And one of the main ways to assess autonomic dysfunction is a questionnaire that is termed the Compass 31 questionnaire, which includes a set of 31 questions. And these are related to different autonomic symptoms. So, for instance, there is a group related to orthostatic intolerance, which is the most heavily weighted. There are other questions related to gastrointestinal function, vasomotor function, bladder function, and so on. So this is perhaps the best way to assess autonomic dysfunction via a questionnaire. So there have been several studies published. I'm giving 2 examples here. And indeed, the post-COVID condition seems to affect autonomic function. And this was reflected on the responses that the post-COVID individuals gave to the Compass 31 questionnaire. So this brings us to our study, which has been funded by the CIHR. We have a great team. Marie-Hélène is part of this team, as is Rick Hogue and a bunch of other researchers that I'll mention at the end. So what we're trying to do here is that we are trying to sort of put everything together in a study that is more comprehensive, and we will use both the physiological monitoring as well as functional neuroimaging, functional and structural neuroimaging, performed at 7 Tesla. Now, using 7 Tesla will allow us to go into a greater spatial resolution when examining the function of these small autonomic centers that lie in the brainstem. So this I think is important in the context of the present of the question that we're trying to answer here. So we're currently recruiting. We aim to recruit around 80 to 100% individuals that do not have post-COVID, are not affected by post-COVID syndrome. And we are aiming to recruit around 160 to 180 post-COVID condition patients. And they will be age-matched and so on. A brief idea about the inclusion criteria. The age group is shown here, 35 to 60. We will use Compass 31 questionnaire to identify subjects, patients that have specific autonomic dysfunction symptoms. So we will require that they respond to positively to at least one subdomain of the Compass 31 questionnaire. So that's an important inclusion criteria for our function. But we will also exclude individuals that have experienced beforehand chronic breathlessness, fatigue, as these are very closely related to some of the symptoms that we see in post-COVID condition. And some other exclusion criteria as regular smokers, individuals taking pregabalin, and individuals having MR contraindications. Now, the general idea of this study and its structure is shown on this slide again. So we will have them complete a number of questionnaires, one of which is obviously the Compass 31 questionnaire. Some other questionnaires also Monse is using in her study, so the breathlessness, fatigue scale, and so on. Most of these will be done online on REDCap, and then all the individuals that will be invited to participate in the full study, they will be required to come to the lab for 2 sessions. One session will be done outside the MR scanner, and then we will, during that session, we will record several different physiological signals, and we will also implement different standard physiological testing that probes autonomic function. And the second session will be done in the 7TESLA MR environment, and there we will collect structural data, as well as we will implement some of these autonomic tasks in the scanner to get a more clear idea about the activation patterns that we may observe in the brain and how these may be different between post-COVID condition individuals and controls. So again, a rough overview, we will try to extract the following markers. There are some, the first set of markers is related to cardiovascular autonomic function. So for that, we will ask the subjects to do a sit and stand test. We will ask them to do a Valsalva maneuver, and we will also ask them to do a cold pressor test. And these will be done outside the scanner. Then we will also probe respiratory and chemoreflex function using the markers that I'm showing over here. And to do that, we will ask them to do a standard spirometry test. We will also ask them to do a slow breathing task both outside and inside the scanner. Okay, and we will also implement hypercapnia, which means that we're going to give them air with a higher concentration of CO2, and hypoxia, which means breathing air with a lower concentration of O2. And we will measure the responses both inside and outside the scanner. Another set of markers is the cerebrovascular reactivity markers, which we will assess both inside and outside the scanner. And these are related to the cerebral blood flow response to hypercapnia and hypoxia. And again, we will do that inside and outside the scanner. And we will also have additional neuroimaging markers that are resting state fMRI patterns, QSM patterns that I mentioned in the brainstem study that I presented before, as well as another sequence of structural MR that is well suited to detecting damage in post-COVID condition. And we will complement all that, of course, with the questionnaire scores and the blood and saliva biomarkers. So bottom line, we will have a bunch of different markers here. And the final outcome of this, what we're aiming to do in the end, is to identify a set of biomarkers that explain symptomatology, the post-COVID condition symptomatology and its severity. So we will hopefully be able to build a statistical model that identifies the most informative biomarkers and how they correlate possibly to different phenotypes of the post-COVID condition, as well as its severity. So that will be our final goal, which I think is important for many different reasons. So yeah, here's, I don't know how I'm doing in terms of time, but here are some preliminary data that give you an idea about how these protocols affect, for instance, the bold fMRI signal. Here we see that, this subject is breathing a little bit more CO2 during this segment. And this is the hypoxic segment where you can see a clear drop in the O2 that they're breathing in and out, and how this affects the bold fMRI signal. And then you can take this data and to identify more focal activations in the brain of both controls and post-COVID individuals. So I think that's this is all I had for today. I would like to thank the project team, the Sophia, Gayung, Junki, Alice, Angel, Remy, and Mary, and my team of collaborators, of course, Marie-Hélène, Rick Hogue, and a bunch of other collaborators, both from McGill and other universities. And with that, this is our recruitment ad. Feel free to distribute as needed. yeah, we're looking forward to scanning more, to testing more people, and better understanding the post-COVID condition. Thanks.
Thank you so much.
Hi, my name is Todd Davenport. Wow, what a wonderful set of emerging data coming out of the lab. So exciting to see. And just sort of gets my wheels spinning as a physiotherapist and exercise scientist, hoping to bring a little bit of rehab clinical relevance to the conversation this morning or afternoon for you all. So I'm coming to you from the southernmost province of California. And so welcome to my talk entitled Infection-Associated Chronic Diseases, Conditions That Break the Rules. I do want to acknowledge my collaborators, Stacy Stevens, Jared Stevens, Dr. Mark Van Ness, and Dr. Chris Snell from the Workwell Foundation. So what are the rules? The rules are you should feel a little better after activity following rest. You should be able to reproduce your exercise and activity measurements from day to day. There shouldn't be wild fluctuations. Your heart rate ought to increase more with more workload, and you should get better with most things with a little exercise. And I want to talk a little bit about infection-associated chronic conditions, including long COVID, because they break these rules. And so the first broken rule is that you should feel a little better after activity following a little rest. And so this is data from folks with ME/CFS, and actually it's from a publication that's now 14 years old. So it's far from groundbreaking new news in Nature. But we published this 10 years ago, looking into the symptomatic responses to exercise in people who are age and sex match controls, all women actually, and individuals meeting the Fukuda criteria for ME/CFS. And what you can see on that top graph, that top figure, is sort of what we would expect from individuals who are non-disabled. So you expect them to be fatigued immediately after the task of completing a maximal cardiopulmonary exercise test. You would expect them to have muscle and joint pain. You'd expect them to have maybe a headache or some nausea. There's a reason we have a bucket in the lab. And then also weakness and instability, trembling. That's very common after a maximal exertion. But the next day, notice that the symptom counts went way down. The controls were basically fine and beyond into a week. In people with chronic fatigue syndrome, myalgic encephalomyelitis, we see people who are fatigued when they first get off of the apparatus, the equipment, but they stay just as fatigued a week later. You see people with lightheadedness that are substantially more. We see this as a new symptom. We also see cognitive dysfunction right after the task. We also see insomnia, sore throat, swollen glands. Some of these symptoms that are present magnify over time. Muscle and joint pain get worse over time. The headache tends to get more frequent over time. The weakness and instability tends to stay. And there are a lot more signs and symptoms the next day and beyond. So very unusual response to physical exertion. Within about 24 hours, we expect about 85% of our sedentary control subjects to fully recover, but no one with ME/CFS does. So the post-exercise, post-exertional response is characterized by a delayed abnormal recovery response. The remaining 15% of our controls are better within 48 hours, and only one of our participants with ME/CFS recovered within 48 hours. And in this study, it took 4.5 days to 2 weeks to recover from a physical challenge in people with ME/CFS. So again, highly unusual response to exertion. And interesting cross-pollination between physical exertion causing cognitive symptoms. We'll come back to that in a minute. This is a results from a questionnaire study by Lily Chu over at Stanford in the southernmost province of California, and her colleagues. And what she did was she looked into various different stressors for post-exertional symptoms and signs, and found that symptoms were more frequent after physical and cognitive exertion than they were after emotional distress, again, suggesting that we're not talking about a major psychiatric condition here. We're not talking about anxiety and depression. This is something altogether different. And, again, multi-symptom presentation: fatigue, poor concentration, difficulty thinking, of relevance to the lab here, but also muscle pain and sleep disturbance, poor memory, again, of relevance to the lab's work. flu-like feelings, joint pains, those are all very common after physical and cognitive exertion. And so trying to disentangle some of these symptoms is difficult because they occur in a constellation, which I think is important to point out. It's really hard also in a research sense to study post-exertional symptoms and signs because of the kind of decoupling of cause and effect. There's a length of time between a potential stressor and the symptom, which may vary, and so it can be very difficult to identify cause and effect unless you're using very specific paradigms to establish that cause and effect relationship. The duration of post-exertional symptoms and signs also can vary, from 1 to 7 days. But again, it can absolutely vary based on the type of exertion, where a person is with their fluctuating baseline, and so forth. So there's a lot of data that should be collected surrounding time course and typical pattern of symptoms and signs in order to really understand the complex pattern of post-exertional symptoms and signs. And obviously, this is data from mixed methods work that we've done. The symptom experience is obvious misery, lower functional baseline, exclusion from desired activities in society, people who feel like they've been in a barrel and rolled downhill or been through a trauma, from a cardiopulmonary exercise test, trouble holding head and body up, terrible headaches, again, highly unusual, very disabling set of symptoms and signs following physical exertion. And so just to make the point that long COVID obviously is more than just tired, fatigue is a very commonly reported symptom and sign associated with long COVID. But this whole idea of post-exertional malaise is really sort of second on the list here, is being among the most common, and tends to predominate over time in the subset of individuals going on to experience chronic long-term disability related to long COVID, and that the prevalence of long COVID symptoms tends to change over time. And the one that notably increases is this pink line here, which is post-exertional malaise, or again, this abnormal recovery response, which increases over time, particularly after about 8 weeks post-infection, suggesting the potential shift in terms of a different phenotype of getting away from the acute condition and more into a phenotype of chronic illness. And this phenotype is really exclusively present in people who have long COVID compared to convalescent controls who are improving, and compared to healthy controls. And so you see again in study after study all of these this multi-symptom presentation associated with long COVID. So again, trying to disentangle these different symptoms may not be serving us well in the clinic. Looking at the pattern seems to be more the way to go here. And just to point out that this abnormal recovery response is so unusual that just asking about cognitive dysfunction, fatigue, joint pain, sleep disturbances, and whether a person's symptoms tend to get worse with activity is a very good way to identify whether people may have post-exertional abnormal recovery response related to exertion. So the second broken rule is that you should be able to reproduce your exercise measurements from day to day. And we know that cardiopulmonary exercise test is reliable to detect small changes in cardiac pulmonary metabolic status between days. So the 24-hour test-retest reliability is very high in healthy controls, trained individuals, athletes, and including people with conditions on the screen who are very ill. So failure to reproduce a maximal test result is unusual. And so the paradigm that we've used in our lab is called the Stevens protocol, or a test-retest cardiopulmonary exercise test. So we do pre-test sets of measures rolling into the day one cardiopulmonary exercise test, which we then repeat 24 hours later to identify physiological abnormalities in the post-exertional state. So really what happens here is that there's a significant reduction in the volume of oxygen consumed in workload at ventilatory anaerobic threshold, which is sort of the transition point between being able to operate without oxygen, the point at which the aerobic metabolism starts to become defeated by its own byproducts. And so what happens is a drop in the ability to function, the metabolic ability to function that we see physically at a systems level. And in fact, as a biomarker, it's 95% correct in identifying people with ME/CFS and 90% correct in identifying matched sedentary controls. And so this is data from, again, not hot off the press, 11 years ago. This is what we've known for a long time, and seems to be the case also rolling forward into long COVID. But it also reproduced in labs around the world and on systematic reviews. And so there really does seem to be some abnormality in carbohydrate and short chain fatty acid metabolism that may be at least partially at the heart of these metabolic problems that we see in long COVID. And of course, the brain has mitochondria. The metabolism is slightly different than skeletal muscle in terms of substrate, but at the same time, thinking about sort of a unifying hypothesis in terms of bioenergetics, it's not totally surprising to me to see changes in brainstem activity that look like age-related brainstem changes, because mitochondrial activity is known to reduce also with aging. We've developed a clinical staging for post-exertional symptoms and signs, and we sort of distinguish between the immediate symptoms and signs that are almost characteristic of the normal response, but out of scale with the normal response, and then short-term symptoms that tend to involve muscle and joint pain, more cognitive dysfunction, headache, and sleep disturbance, and then long-term symptoms that seem to reflect a more sustained immune response, more of a sustained damage to the metabolic system, particularly the aerobic energy system, including weakness, decreased in function. That's where you see flu-like symptoms and all of these cardiopulmonary symptoms that may be reflective of dysautonomia. So the third broken rule is that your heart rate should increase with more workload up to a theoretical maximum. And unfortunately, we don't see that. We see what we've termed chronotropic incompetence, which means that the heart rate goes up, but not enough, according to age predicted. And in fact, the dysfunction seems correlated with the extent of self-reported disability. So people who have none to mild tend to have less chronotropic incompetence compared to people who are in the moderate to severe category. So again, a lot of what is seen in terms of dysautonomia in response to orthostatic challenge, we also see in response to exercise. And this may have to do with a failure to withdraw of vagal input, and because that's necessary in addition to sympathetic input predominating to raise your heart rate during exercise. So there may be some dysfunction there, and so this might point to the cholinergic dominance hypothesis that Michael Van Elzacker and his group put forward, suggesting that a vagus nerve infection triggers an exaggerated sickness response and immune response. So the last broken rule I'll talk about before I stop and open the floor to questions is that you should get better with a little exercise. Physios have really, in particular, created this exercise as medicine and promoted this exercise as medicine idea. And so, a lot of things get better. If you're down with a virus, it's good to kind of get up and take a walk and so forth. And here's why that's not a good idea. You have a broken aerobic energy system, at least according to our assessment of the systems level response to physical exertion. And, Black and McCulley back in 2005, again, not exactly hot off the press here, everyone, found that they were able to get people to do a graded exercise task involving an increase in step counts. But when they came back and reanalyzed the data in their second publication, what they realized is that the increase in accelerometry counts was not related to an improvement in a person's ability to do daily activities. It was all about just doing the exercise. So the graded exercise turned into a self-fulfilling prophecy. So this points to another caution in research is when we're looking at naturalistic data related to a person's activity level, we have to be very careful that we're not just sort of measuring what it is that we induced based on the intervention that we prescribed, and that there is a fluctuating physiological baseline of post-exertional malaise that we have seen in terms of a lack of reproducibility in test-retest performance on cardiopulmonary exercise testing. So that some activities based on their metabolic level are going to be more difficult when a person is in a post-exertional exacerbation or a crash. And we can't assume that the system will adapt normally to chronic exercise if it does not have a normal acute exercise response. So here again, there are a lot of physiological reasons why just taking a walk is not a great idea. I always like to bring up patient data. Patients lead us. And so listening to patients is obviously something that researchers and clinicians ought to do. Graded exercise, according to one large survey of the ME Association in the UK, was the most likely to worsen and the least likely to be considered helpful, whereas pacing was the least likely to worsen and most likely to be appropriate for a person's needs. So again, less is more approach. And the good news is, because I know I'm speaking with an audience of mixed researchers and clinicians, physical therapists, occupational therapists, does not seem to matter. The specialization of the therapists, as long as there is a dedicated approach to pacing. So in conclusion, again, not all exercise tolerance can be fixed with exercise. There appears to be damage to aerobic glycolysis, which may generalize to other tissues in addition to the systems level analysis that we're able to do with cardiopulmonary exercise testing. And this is not deconditioning. That there's impairments in mitochondrial functioning that have been implicated in post-exertional symptoms and signs, myalgic encephalomyelitis, as well as long COVID. And so the primary treatment really is sort of an energy first aid approach of reducing allostatic and chronotropic loads, as well as reducing oxidative stress and trying to support metabolic functioning, which again may be a future direction in research. So I'll conclude with the safe rehabilitation principles that long COVID physio and others have really put forward in terms of first excluding exertional oxygen desaturation when working with patients. Then assessing for post-exertional malaise or post-exertional neuroimmune exhaustion, assessing for dysautonomia and orthostatic intolerance, excluding cardiac impairments such as through myocarditis, before beginning with a trial of stopping, resting, and pacing, which is again considered the safest place to start in folks with long COVID. So I think I'm done. So I really appreciate your attention and hanging with me with the speed of the talk.
It was perfectly on time. Thank you so much.